similar that we actually treat all the fluid present in those 74 trillion cells to be one fluid present in one compartment or one cell. So very similar fluid in all of our cells. So intracellular fluid compartment is one. Then we have extracellular fluid compartment, which is really fluid present everywhere outside the cells. Wherever it is present, that all collectively is extracellular fluid. Extracellular fluid compartment then in turn is divided into intravascular, as we saw here. So this is intravascular, and interstitial, which is here, interstitial. So intravascular and interstitial, they both are part of extracellular. There is actually one more extracellular compartment which is called transcellular. There is one more which is called transcellular, but usually not really studied as part of the body fluid compartments. It's pretty stable. About 2 liters of the whole fluid is present in the transcellular. What that is really the fluid present in our joint cavities, the fluid present in the pericardial sac, the fluid present in the peritoneal sac, CSF, and so on. So that fluid all collectively is called the transcellular fluid. Usually not studied within the context of the body fluids, just because it doesn't change that quickly. But the remaining fluids would be, you would see that they would be shifting and increasing and reducing. So transcellular is also part of the extracellular. Another very important thing is that the composition of the transcellular is different from the composition of the other two compartments for the extracellular. So that is a dissimilarity. Okay, so these are the basic compartments which we have. Now, interestingly, so as we just established, this whole compartment is extracellular, but within that we have two subcompartments. So the question becomes, why do we have to further division inside these compartments? The reason for that is that the blood vessels, so I can actually just go ahead and make this look like a blood vessel. So let's say this is a blood vessel. So the blood vessels and the fluid present inside the blood vessel has a different composition as compared to the fluid present inside the remaining interstitial space. So that is why we separate the extracellular, or we divide the extracellular fluid into two primary compartments, intravascular and interstitial. We'll talk more about what is the composition differences between them. Now, as a medical student, as a doctor, as a nurse, you have to understand one very important thing. What keeps the fluid in a particular compartment? So let's say that if I say I have 3 liters of plasma here, you should be able to tell me why that plasma, why that fluid does not just spill into the interstitial compartment and drain the blood vessels dry, or reverse. Why does all the interstitial fluid not move into the blood vessels and just stay in there? What keeps it separated from here to here? Similarly, you would notice as we progress further in our lecture that the fluid which is present here usually does not just move out. It's not that the cells would just collapse and all the fluid would move out and start sitting in the extracellular fluid. So what keeps the fluid in here? So that is the most important thing, that how does the maintenance of volume happen, number one. And of course from there then we would talk about what causes the shift in those volumes. Will there be a situation where the volume here would reduce? What will cause that? Or will we have a situation where the volume here will increase? What will cause that? Will we have a situation where the volume here would reduce or increase, and what would be the factors? What will be the factors which would affect the volume here? What would be the factors which are keeping these compartments intact as they are? So these are the important things which we have to understand. So let's start first of all from the very basic thing, that where do we get water from? So I want to establish one thing. Usually the intake brings the fluid into the vascular compartment. Usually the fluid is added to the vascular compartment. From here it will move into the interstitial compartment if it has to, and from there it will move into the intracellular compartment if it has to. Similarly, when the fluid is ejected, I'm talking about physiological situations, normal physiological situation, normal healthy person. I'm not talking about pathological situations. I'm not talking about denudation or of the skin, or I'm not talking about the damaged viscera and so on. I'm talking about normal physiological situation where a healthy person, we need to know how the water is added to the body, and we need to know how the water is removed from the body. So the water removal is also going to be working through the intravascular system. So of course there are organ systems which are attached here. For example, for the injection or intake of the water, we have the GIT. Similarly for the exit, so let's say this is the intake. Similarly for the water loss, we would have some organ system. I think you are aware of the kidney. That is a system we are actually trying to study, and this is the context we are establishing for the kidney. So the renal system is one of the systems which helps us lose water, or helps us regulate the amount of water by deciding how much water to lose. So the water is coming into the vascular system, water is exiting from the vascular system. The interstitial fluid and the intracellular fluid are then going to respond to the changes which are appearing here. So this is a very, very important concept to understand, because I've seen so many times that, remember the Don and Yarrow diagrams which are something like this. Let's say this is the volume, this is osmolarity. So let's say the osmolarity is about 300 milliosmoles, volume is, let's say, 42. Then we say that, all right, the volume of intracellular fluid, that is this representation over here, and then here, let me make it correct, then the volume of the extracellular fluid, that is this compartment represented here, which then is divided into intravascular and the interstitial compartment. In this diagram, you would see many times that the questions which come in USMLE and in our general hospital practice are, what will cause this, let's say. So in this situation, what you're seeing is osmolarity has increased, volume has decreased. So how would that happen? And many times what I've seen is that the students try to understand like this or try to memorize it. But really, if you understand this, that the volume changes would start rippling in a normal case from the vascular system, and they would then ripple into the interstitial and then ripple into the intracellular. If you remember this, so start from here and then move to the other sides. So that is one major concept. So going back to this, how do we get the volume? How do we get the fluid to come into the vascular system? So how do we get water? So there are two primary sources of water. So one source is the water intake. So that is the drinking water which we have. So let's say here is a guy who has been drinking water. So that water which he drinks is going to go into the GIT, and from the stomach and the intestine, it's then going to be absorbed into the blood vascular system. From the vascular system, then we'll see that it can go to the other compartments if need be. So that is one way of water intake. So we have about 2100 milliliters per day of water intake. But this is not really hard and fast. It depends upon the person, it depends upon the temperature, weather, situation. So it could be more or it could be less, but again we're talking about the average situation. So about 2100 milliliter of water intake per day. So do not forget these. We are going to use all of these concepts as we build up on the body fluids. Then we are going to use them in two primary areas. We're going to use them in the renal system, and we're going to use them in the cardiovascular system. So be very careful for what we are working through. Okay, the other way of